All right, so we're going to do some practice today on how. So yesterday we talked about, not yesterday, but when I saw you last, we talked about traits and genotypes and phenotypes. And so today we're going to practice using a grid called a Punnett square. And we're going to, let me pull my share screen up just for a minute and then come back. All right, so the Canvas unit that you're pulling up is called monohybrids and hybrid crosses. So this is an example of what we're going to practice today. So look at the first box and um, this is what you're going to be expected to know how to do. So in the little quiz that you did the other day, they asked you which traits were dominant, which ones were recessive, which were homozygous, which were heterozygous, and so on. So when we're looking at pea plants, yellow is dominant, and they use the capital Y to represent the dominant trait. So in this box over here, and I don't know whether you're seeing my arrow on the screen or not. Do you see my arrow moving on this on the screen? Is it showing for you? In any case, I'm looking at the yellow box and it has two capital Ys, and that represents one of the parents of the pea plant, and they have both of their genes. Remember, we have two sets of genes. So one chromosome has one yellow and the other one has another yellow, but they're both dominant on this pair of genes. All right. Then we have the other parent who is green, and green is recessive. So we have lowercase y's. Okay. Now, again, during meiosis, remember that there were four um, egg cells produced during meiosis and four sperm cells produced during meiosis, and actually one egg. But we think about the possibilities that can go. Okay, let me back up. There are four cells that are created during meiosis for the female. Only one becomes viable. But there are four cells created during meiosis, and then the other three just kind of go away. And there are definitely four sperms created. And in each of those, you're going to have one Y, one capital Y, or one lowercase y. So that's what it's showing you here, the gametes. Okay, so it's possible from the yellow one to give off only capital Y. So it doesn't matter what gametes you have, they're only going to have capital Ys. In this case of the green one, their gametes are only going to have, yes, I'm sure you did. If you did life science in seventh grade, you probably did do Punnett square. So this so good. This will be. Does everybody remember how to do a Punnett square? Okay, so this should be quick, quick, fast, and in a hurry. We won't have to spend a lot of time with it, but consider this a review. All right. So remember, the first generation is called the P generation. Those are the parents, and when we cross these two, we create the F1 generation or first filial generation. And notice, because one is giving a capital Y and one is giving a small y, the only characteristic that the offspring in the first generation can have is big Y, little y. All of them, they don't have a choice. All are going to be capital Y, little y. So they're all going to be heterozygous for the dominant trait, which happens to be yellow. Okay, so all of them will be yellow. Then what we do is take two of those and take the gametes from those. And so what they have to offer, all of them have to offer, is one capital Y or a lowercase y. And we put those on the Punnett square. So we see now again, the Punnett square represents the possibilities. We have a 100% possibilities, and we divide that into fours. So it's 25% in each box that we're going to look at. So we look at percentage and possibilities. We're going to talk about ratios, and we're going to talk about genotypes and phenotypes. All right. So what we do with the Punnett square is we put one parent across the top and another parent across the side. And then we donate one allele from each parent. So in the first box, we take one from the top and one from the side, and there just happen to be both capital Y's. So in that first box, we have two capital Y's. In the second one, we take that one green and we match it with the capital Y on the other side. So in this box, we have a capital Y and a lowercase y. Okay, so we're doing the first row with the two possibilities from the top and the side. They're still yellow because yellow is dominant. So capital Y and small y are still yellow. We do the same thing with the box on the lower level. We take the Y from the top in that first column, and we take the small y from the bottom box on the side. And in that first lower box, we have a capital Y, lowercase y. Then we take the lowercase y from the top and the lowercase y from the side, and we put that in the fourth box, and both of those are lowercase y, so that one will be green. So in the second generation, you don't have 100% heterozygous yellow. You have a variety, okay? Now there's two ways to look at this. We can say that we have 3 out of 4 are going to be yellow, and 1/4 is going to be green. We can say this is 75% yellow and 25% green. Or when we look at it in terms of the genotype, we have 25% homozygous dominant yellow, homozygous yellow. We have 50%, because we've got two boxes, of heterozygous yellow, and 25% homozygous green. Okay, so that's a couple of ways that we look at reading that box. Okay, all right. Now, this, when we say a monohybrid cross, we're only looking at one characteristic, but we know organisms don't have one characteristic. On a chromosome, told you to think of a chromosome as a string of pearls, and each bead is a characteristic. So if we were looking at this, this would be on a chromosome, and only one of the beads would be for pod color. But in real life, there are several characteristics on a chromosome. So even looking at a dihybrid cross, like the one on the next slide, is only looking at 2 out of all the possibilities that are on that chromosome. But you're going to do these the same way. Whatever the one parent has, you're going to represent in one circle, and in the other parent. So in this case, we're looking at not only color, but we're looking at the shape of the pod. So one is smooth and one is wrinkled. Okay, and since or round and wrinkled, so they're using R, so we're going to assume that the round, R is for round. So we've got yellow and round are dominant, and green and wrinkled are recessive. All right, so again, we're going to put one parent at the top. But this is where it gets tricky, because then you have to separate them out in pairs. So you're looking at, what is one gamete going to have? And again, the gamete is looking at like a pearl, string of pearls, and that chromosome is going to have 2 of the different ones. So it's going to have one pearl is going to be, or say one bead is going to be for the color, the other bead is going to be for the shape. So you're going to have a Y and an R. The other possibility is to also have, okay, the first one at the top that is going down the side. So you're going to have 2 capital Y's, and it should be, yeah, and two capital R's are a possibility. But you can also have, okay, I see what they did. All right, let me back this up, because I'm skipping. I'm looking at the box and not looking at the one in the middle. I skipped the whole first generation. All right, so again, if you have a pure heterozygous, so this one is yellow and round, and they're both dominant, and this one is green and wrinkled, and they're both recessive, then the only possibility for your offspring is to be heterozygous, yellow, and round, but you're carrying those recessive genes for um, green and wrinkled. Okay, so when you take this first generation and you take two of them, so that's what this chart is at the bottom, taking two members of the first generation and creating a chart at the bottom, then you're separating these out into pairs so that you have a Y and an R and a Y and an R and a Y and an R on both sides, and the same thing going down the side. So then you're going to do the same thing you did with the first chart, except now you've got 16 boxes instead of 4 boxes. Okay. So you take one from the top, one from the side, and you put that in the first box. And then when you put them back in the box, then you put your letters back together. So like we segregated, and then remember we have some laws that we need to talk about, laws of segregation. So we segregated out all of the characteristics so that one gamete has a Y and an R. The next gamete has a Y and an R, but a lower Y and a capital R. The next gamete has a capital Y, a lowercase r, and so on. So inside, we have a fertilized situation where these two have come together. So in the first box, we had a capital R and a capital Y. On the side, we have a capital R, capital Y. So in that first box, that's how we put the Y's back together and the R's back together, and we have um, capital Y, capital Y, capital R, capital R. Then we stick with that first one in the first row, and then we go all the way across on the top. So we take the lowercase y and a capital R, and we match that up with the capital Y, capital R, and we put that in the next box. Okay? And then we go back up to the top again, and we take that capital Y, capital lowercase r, and we're again matching it with on the one on the side with the capital Y, capital R, and in that box we're getting capital Y, capital R, lowercase r. Okay, is everybody following? Because you all said you've done this before, so is everybody understanding how we're putting these in? Because if you understand how to do it, then I don't have to spend. Is there anybody who doesn't remember how to do a Punnett square? Or everybody who's not understanding what I'm doing? You just put it in the chat. I'll keep explaining if you need me to. You understand the second box now? Okay. So again, we've got vocabulary. We've got monohybrid, which we were only looking at, think of mono, look at prefixes. A lot of these things are about understanding the prefix, homo and hetero and mono and di. Mono means we're only looking at one characteristic. Dihybrid, we're looking at two di means two, so we're looking at two characteristics. And eventually you look at a polyhybrid cross where you're looking at several characteristics. But for now, we'll just stick with these two. All right. So that being said, let's go back to I'm going back to Canvas now. So hopefully you left your Canvas up. All right, so what you need to remember about doing a cross is that we are predicting possibilities. It is possible that this egg and this sperm, which has these genes, are going to combine to produce these offspring. All right, so let's go through. All right. The main idea behind predicting traits of offspring is probability. So again, the Punnett square is a probability box. It's like, and we're looking at the likely outcomes for a given event. Traits that are passed on from parents to offspring are determined the combination of genes or the genotype that are in the gametes, which allele is passed on by the parent is random in nature. So probabilities of a particular event occurring is a fraction of the outcomes in which event occurred. Again, when we say probability, we have this box and saying that this is what's going to happen 100% of the time, but it might be in nature. The first boxes might be the one that occurs all of the time. All right, we might always get the heterozygous version. We might always get the homozygous version. We might always get the recessive version. So this is a probability. We can predict what can happen and what cannot happen. That's what this does. Okay, so they compare it to a coin toss, and every time you toss the coin, there's only two sides to a coin, so you have a 50-50 percent chance of getting one thing or the other. But every time you flip that coin, you could get heads every time, you could get tails every time. 5 out of 6 times, you can get heads. It's a thing of chance. Okay, so all we're looking at when we do Punnett squares is probability. This is the probability of what can happen. All right. Um, Sex chromosomes. So last term we talked about that there are 46 chromosomes in the human body, 23 pairs. One of those pairs is coded just for maleness and femaleness. All the rest code for whatever other characteristics that you have. But the chromosomes that you have for XX or XY code for all your female characteristics or all your male characteristics. Okay. All right. So again, you need to remember that the female has two X chromosomes, male has an X and a Y chromosome. And during what we call segregation, when they split and go their separate ways, it is possible that the mother can give one X and the father can give one X. In this case, you get girls. If the mother gives one X and the father gives one Y, then you get boys. And so you don't know how often that's going to happen. But the probabilities of getting a girl is higher than getting a boy, but it still comes out 50-50 if you do a Punnett square. I'm not going to read that, I'm just explaining that. All right, so everybody said they are clear with this. Again, remember that we are looking at alleles, and alleles are a particular trait. Okay, particular trait. So on your chromosome, you might have eyes and height and hair color all on the same chromosome. So those are individual characteristics. And again, the X and Y chromosomes are going to have all your characteristics for being female or being male. So those will be on that particular one. All right. Okay, we've talked about this when we did our example. Okay, these are, and I'm going to give you a test cross worksheet in just a second. All right, so they were talking about when the one that we did already was color and shape, but then sorry, pod shape, pod color and pod shape. And now we're looking at the flowers. So the flowers work the same way. We're either talking about purple being dominant and white being recessive. And then notice in this one, they used the capital letter B. She did say it was random. Now, when you do these activities, they will always tell you which one is dominant, and they might even tell you which letter to use. So in this case, they are using a capital B for the dominant trait of purple, and they're using the small b for the recessive trait of white. But bottom line is capital is always dominant, lowercase is always recessive. All right, so here's another one, a dihybrid where they're looking at, what are they looking at here? The tail. So you either have a long tail or a short tail, or you're looking at the color. Okay, and there's that again. All right, so we need to talk, oh, we didn't talk about the ratio. So with the ratios, you are counting how many you have in the box. Ratios are easy to do. For example, and we're looking at who's hybrid who's pure. So you're going to look at there's three, maybe three dominants, three recessive, one pure. So you in this box you have uh all the boxes are going to add up to 16. So yeah, 15, 16. All right, so you're counting all of the individuals in the boxes and they've color coded it for you, which they do sometimes, so that you know which ones are which ones they're referring to. So you have three boxes that have the what is it? And this is so small. Let me make it bigger. Zoom a little. All right, there we go. Now I can see. All right, so you've got three boxes that have um, the two capital S's and two small B's. Where's the other one? One, two, three. One, two, three. So they're going to be white with short tails. There's three of those. Then you have three that are going to be brown with long tails. And then you have 9 of them that are brown with short tails, and only one that is both white and has a long tail. Okay, so short tails are dominant, long tails are recessive. No, then let's see, I said that the wrong way. White for brown, yeah. Short tail, yes. That's short tail is dominant. Long tail is recessive. That's what I said. Okay. All right. And again, they will tell you which is which. So you have to be able to take this box and look at this and be able to read it out and give the ratios. You need to be able to say the phenotypes and the genotypes. So the genotypes are the big S, big B, little s, big B, um, big S, little b, and small s, small b. Those are the genotypes. You're telling me what genes are present in the box. Phenotypes are telling me what do they look like? What am I seeing at this individual? Um, so I'm going to let you work on this practice grid while I do attendance. Whoops, not that one. This one down here. Practice on that for a minute, and then I'm going to upload a worksheet for you to work on after I do attendance. Okay, we got quite a few people missing today. And if you need to read over that page, by all means, please read it over. Okay. Ariana, you're doing math and you're not doing biology. Thank you. Um, okay, that's done. Okay. I'm trying to share the assignment page with you all, but it is taking its sweet time. Okay, there it is. Mm, all right. I'm having trouble getting the worksheet to be accessible to you. So I have to edit the page again. So you might see two links on the page. We'll see which one works best for you. Mhm. Try it one more time. There we go. Okay, I can delete this and save that. Okay. All right. So let's try this again. All right. So you might need to refresh your page so that you can see the assignment because it'll come up on your to-do list. It should say Punnett Square practice. Refresh your page in order to see it. Um, Try to view this as a student. Okay. I do apologize. My computer, I need to get rid of some of the files that every time you all send me stuff, and sometimes I have to download it to open it, and I need to get rid of all of those files off my computer because it is slowing my computer down so much. But it should look like that. Let me know if you can open it. Mine is taking more than a minute to open. Why is it doing that? Not able to edit. Okay, so let me save it a different way. All right, so let me stop sharing. It's when I start sharing that the computer does a slow thing. All right, so let me. So which is the way that you can edit the file? Because I was trying, I don't want to send it as a PDF. If I send it as a PDF, can you convert it to your Google Doc so that you can write on it? You put it in Cameo. Okay. All right. Well, then I'm going to save it as a PDF. And then we'll send it that way. Okay. Okay. Hmm. It's gonna be one of those days, snow and everything else. Now my computer is just totally blacked out. Okay, let me close that. Let me edit this. It's Friday. Okay, so I'm gonna take that out and I will. Okay, got it. Okay, so let's see what we have here. Okay, so if you would refresh your page, I put it up as a PDF. And let me know if you can open it now. All right, so when you open it, it should look like this. Thank you, Piper. All right, so if you would work on that for the rest of the time and then upload it when you are done. All right. So again, it tells you at the top that that inflated is a dominant allele. So you're going to, if you like, you can create the letter I as inflated and let the lowercase I be constricted. Okay. So it's okay for you to pick a letter, but it needs to represent the dominant allele. On the second one, um, what, what is yours saying? I have to blow that up so I can see what that says. And it might pop up on the screen, folks, so give me a minute. Actually, and folks, sometimes y'all just need to come off mute and just tell me what your problem is, because right now it's taking time for me to blow up what you sent me. And I can't see what that says, error while processing. Um, okay. Um, all I can say about that is to refresh your page. Anybody else having an error? Getting an error message? Refresh your page, try it again. Oh, don't do the ODT file. You're not going to get that one. So refresh your page, go back to the new document and upload, thank you, Piper, and download the PDF. Okay, thank you. Okay. See, while I kind of work for your school, I'm not really in their system, so I don't have some of the privileges. When I send you stuff, I have to figure out how to convert it to you. The same way when you all send me stuff, sometimes I have to ask you for permission to open it. So it's kind of tricky. All right. Anybody else having a hard time? All right. So what you want to do is create it so that you can type in it and then type in. So remember, genotypes, you're typing in what gene. So like I said, for decide ahead of time what letter you're going to decide for the dominant characteristic and let that be the letter that you use and the lowercase letter. So in other words, you're not going to have an I and a C. Okay, that's going to be wrong. You would have a capital I and a small case I if you're going to use I as inflate. So give yourself a key, and that's what's going to be in your genotype. So tell yourself what letters you're going to use. And again, if I'm going to say for this chart, the possible genotypes for the first one are capital I, which would be a inflated pea pod. Or I can have capital I, lowercase I, and that's still an inflated pea pod. So that's your phenotype is inflated pea pod. If I have lowercase I, that's a constricted pea pod. So that's how you're going to answer that box. In the first column, you're going to put what are your letters? And in the second column for phenotype, what does it actually look like? And you're going to do that with the first two at the top. And then so it's kind of like 1 and 5. And then for 2, you're taking your inflated homozygous constricted plant, some homozygous inflated and a homozygous constricted pea plant, which means you're taking whatever you wrote for the first 2. So it's going to be capital I. So if we're using I, it's going to be capital I and capital I crossed with small I, small I. So that's what number 2 is saying. Okay. All right. So any questions about what you need to do with this? Are we all in agreement? We're going to use a letter I. Down here, you can do what they've been doing using P for purple and capital P for purple and lowercase p for white and so on. Okay. All right. So I'm going to stop talking, let you work on that. And again, when you finish, just upload it to the assignment page. If you have a question, definitely ask in the chat or come off mute and ask it out loud. Again, if you have any questions, please ask. Temperature is dropping. All right, so if you know what you're doing, you just work on that over the weekend when you get bored. And make sure you get that loaded back up by Tuesday. But if you have no questions for me, you are free to log off and you have a safe and wonderful weekend.